everyone, my name is Britni Livar. I'm from the newly established School of Ocean Features at Arizona State University, and today I will be talking about the role of particle-associated bacteria in aggregation formation in the ocean. All right, we'll start off with a quick overview of the biological carbon pump. Now, the biological carbon pump consists of the photosynthetic fixation of inorganic carbon into particulate and dissolved organic carbon by phytoplankton. This organic carbon can then be exported to depth by such means as marine snow or fecal pellets. Specifically, we will be focusing on the formation of aggregates by bacterioplankton. Live video that will zoom us in to Bermuda, located in the Sargasso Sea and off the coast of the US. Now, the Sargasso Sea is located in the North Atlantic subtropical gyre and is considered an oligotrophic ocean region. Clays from the Saharan Desert are deposited within the sea by trade winds, and these additional particles can act as ballasting material, and they can also influence aggregation. Our research specifically focuses on heterotrophic marine bacteria, and heterotrophic bacteria have been previously studied in association with phytoplankton, wherein TEP production, or transparent exopolymeric particles, and aggregation formation was found to be influenced variably, but rarely have the role of heterotrophic marine bacteria in aggregation or their aggregation potential with each other has been the sole focus of such studies. Therefore, my work with Pseudothermomonas carrigenivora and Vibrio thalassi, two bacteria selected based on an indicator species analysis and their specificity to sinking particles, alongside Marinobacter adherens HP15, a bacterium found to attach and enhance the aggregation of diatoms, is contributing greatly to this field. I would now like to introduce transparent exopolymeric particles. So I previously mentioned them, but transparent exopolymeric particles, or TEP, are sticky acidic polysaccharides that can act as a glue to hold together aggregates. Through this, TEP can increase the size and density of particles formed from smaller plankton by incorporating more cells and minerals. Additionally, only some phytoplankton and bacteria exude TEP precursors, and this can vary down to the species level. All the bacteria used in our experiments have previously been found to exude TEP as shown in the figure here, wherein the bacterial produced TEP was stained with Alcian blue, giving it that nice blue color. Moving on, I would also like to talk about the difference in aggregates that I'll be talking about further down the line. The two types of aggregates I'm going to focus on are microaggregates and macroaggregates. Now, microaggregates have also been known as suspended aggregates and are categorized as 5 to 60 micrometers in diameter. These microaggregates require the use of such equipment as Coulter counters or flow cytometers to properly quantify. There is an example image of microaggregates below with a scale bar representing 20 micrometers. Macroaggregates, also called visible aggregates, are larger in size than microaggregates, ranging from about 0.1 to 0.5 millimeters on the low end. They are most likely formed from microaggregates, and these aggregates are analyzed using dissecting microscopy. Now that everyone has a little bit of background on our topic, I'm going to move forward into our objectives. Our first objective is to determine whether particle-associated heterotrophic bacteria can form micro or macro aggregates. And if yes, can we discern the sinking rates of the aggregates that formed? Approach. To share the answer to that question with you all, I first have to go over our approach and our experimental design. So we initially grew the bacteria in DIFCO 2216 marine broth until the end of their exponential phase, then inoculated them into roller tanks at a cell concentration of 10 to the 5th cells per milliliter. Each set consisted of 5 tanks. 3 of the tanks contained bacteria and kaolinite clay that acted as a ballasting material. And the other 2 tanks were controls which only contained bacteria. These tanks were sealed and allowed to roll for 7 days before takedown. Any macroaggregates that formed were carefully removed and either photographed using a dissecting microscope to determine their diameter or placed into a glass-graduated cylinder to determine sinking velocity. Samples were also taken to analyze microaggregate formation using a Beckman-Coulter counter. In our next slide, we will have a video of the roller tank themselves. So you can see them slowly rotating in the roller tank. And what we want to achieve is we don't want the aggregates to fall to the bottom of the tank. We want them to be constantly suspended so that there's no interference from the hard walls of the tank themselves. We are simulating an open water column. So we've done an introduction, we've gone through our approach. Now we're going to move into the results that we received. Before going over the results in detail, I must first give a disclaimer that despite the extensive measures taken to ensure sterility, the first set of roller tanks for Marinobacter adherens HP15 had to be reran due to a fiber contamination, and the second set is indicated with an asterisk after the name. So macroaggregates were found to only form in roller tanks with clay. So none of the results I'm going to show so far were obtained from control tanks. On the left is a graph showing the macroaggregate abundance between bacteria, and on the right is the diameter size in millimeters of the macroaggregates. Pseudothermomonas carragenivora is displayed in red, Vibrio thalassii in blue, and Marinobacter adherens HP15 in purple. The macroaggregates that formed were light-colored and varied in appearance from fluffy to dense. Each roller tank resulted in 2 to 10 macroaggregates that formed, although there was no significant difference in abundance between the bacterial species. Vibrio thalassii had a significant smaller diameter for its macroaggregates compared to Pseudothermomonas carragenivora, but not Marinobacter adherens HP15, as shown here with the significant indicator. The significance was calculated using a t-test with a p-value of less than 0.05. Once it was established that these bacteria could form macroaggregates, we wanted to know the sinking velocity of those aggregates. So it was found that the sinking velocity of bacterial aggregates ranged from about 200 to 1,200 meters per day, which is highly comparable to the sinking velocity of phytoplankton-derived aggregates, which range from 100 to 700 meters per day. So this graph here shows the sinking velocity of the 3 bacteria in meters per day, and the legend staying the same as previous. Vibrio thalassii macroaggregates had a significantly slower sinking velocity than Pseudothermomonas or Marinobacter, around 300 meters per day compared to 700 meters per day. And it is also shown with the significance indicators here. So this connects to the idea of abundance and size influencing sinking velocity. Since Vibrio thalassii formed more aggregates from the same initial concentration of particles, the resulting smaller diameter and therefore slower sinking velocity logically follows. This is all important information on macroaggregates, but what about the microaggregates? In terms of microaggregates, none of the control tanks had a significant difference in microaggregate abundance or volume before or after rolling, and so the graphs were not shown here. On the left is a graph of microaggregate abundance, wherein the number of particles per milliliter was measured, and on the right is the microaggregate volume concentration, shown as 10 to the 6 micrometers cubed per milliliter. The before rolling results are coded blue and the after rolling is purple. Here, both the abundance and volume for the clay tanks did show a significant decrease after the rolling period, with the exception of Marinobacter adherens HB15, which did not experience a significant decrease between rolling periods due to a high level of variability. So here's the big picture. For the first time, we found that yes, particle-associated marine heterotrophic bacteria ballasted with clay can form macroaggregates with sinking velocities comparable to those formed from phytoplankton. And we found that the aggregation of bacteria plankton provides a mechanism for their incorporation into larger aggregates and their contribution to export flux. So in conclusion, it was found that these co-cultures of bacteria and kaolinite clay particles could form microaggregates, likely through such means as TP precursor exudation and the cation exchange capacity of kaolinite clay. Furthermore, these microaggregates were then scooped up to form macroaggregates as shown in this figure here. So going forward, I will be looking at how increasing temperatures and ocean acidification affects aggregation of natural seawater communities. This past May, I was lucky enough to head down to the Bermuda Institute of Ocean Sciences, or BIOS, and I was able to use their mesocosm facilities to simulate both ambient and future ocean conditions. After these conditions were simulated, I transferred that liquid of the natural ocean water into 5-liter roller tanks to observe the effects of those conditions on abundance, size, and sinking velocity of macroaggregates. And what we found is that in ambient ocean conditions, the abundance of macroaggregates was significantly higher than the abundance of future ocean aggregates. However, the size or diameter of those macroaggregates were not significantly different between the conditions. Unfortunately, due to the very fragile nature of the aggregates, I was unable to obtain sinking velocity results for the future ocean condition, but we were able to obtain it for the ambient conditions, and it was found that the sinking velocity ranged anywhere from 500 to almost 2,500 meters per day. And then as I finish out the rest of my PhD, I'm hoping to look at how this affects both the microbial communities present under these conditions and any effects on the TP composition and exudation. I would like to thank everyone in the Neuer Lab, particularly Katrina Shurtleff, Kayla Tran, and Suzanne Neuer. Thank you.